Hello to everyone. Good morning to all. Thank you for your attendance. My name is Lorena and I have the pleasure to conduct this meeting today. In this webinar, we have Dr. Danilo Viana presenting. Dr. Viana is medical geneticist, graduated from UFMS, and he has also a master's degree and PhD in medical genetics from University of Campinas. He is one of our medical doctors at Mendelics, and he's going to talk about next generation sequencing as a diagnostic tool in oncology.
Good morning, everyone. It's for me, it's a pleasure to be here, and I thank you for your time and your attendance. Okay, so the idea of this talking, we had some time ago discussing some cases with our colleagues in Colombia and also in Brazil. And one thing I noticed is that there's some confusion between NGS testing for hereditary cancer and tumor sequencing. So the idea today is to present NGS sequencing, talk about the technique, and discuss the applications of this technique. So when we think about genetic tests, genetic tests uses are for three main purposes in clinical medicine. The first one is for diagnostic, when we provide the diagnosis of the disease or provide the disease classification. The second one is prognostic, when we provide the prognostic information on the outcome of a disease. And predictive, when we predict the response for a particular treatment or predict the future risk for cancer in asymptomatic people. And it has been a long time since this has been in use, since the 60s, with the Philadelphia chromosome in myeloid leukemia, chronic myeloid leukemia. And other techniques have evolved, and the evolution, in fact, was great. The karyotype in the 70s and the Sanger sequencing, and then PCR and FISH and automated sequencing and CGH and MLPA, and then NGS. All these techniques has their advantages and disadvantages. I won't go into specifics of each one, but it is important to consider all of them. And when we want to use a test, we simply need to think what we're looking for. If we're looking for something like a big translocation, so I don't need to use necessarily FISH for detect this translocation. I could use karyotype, standard karyotype. But if I need to detect amplification of HER2 in a sample of breast cancer, so I can't use karyotype, so I need here to use something that will be able to detect what I'm looking for. And I can use two probes in this FISH technique and compare the ratio of one probe to the other. And this is how one looks for HER2 amplification. But if I change the disease, I now need to know the mechanism of the disease to look using the same technique for this other disease. So if we want to find ALK fusions in lung cancer, for example, we need to use two probes in FISH, but these two probes will work in a different way. We're looking for a break apart. If a fusion occurs in ALK gene, then we will see these two signals of these two probes to split. And this would be my positive test. So I need to know what I'm looking for. I need to know the mechanism of disease. There is no test that is simply better than the other. And if I add another technique, in this case, I'm adding CGH, it's not that CGH it's better. It's simply that if I want to find a 4P deletion and I could use karyotype, but maybe it will be faster to use FISH. But a CGH seems to me in this situation a big shot, so it wouldn't be needed. On the other hand, if I have a family with typical hereditary breast cancer and I have made all tests and I couldn't find the cause, then I might use CGH to try to find any alteration in genomic composition to see if I can find a cause, and this would be a good use of CGH. And so we have many examples of genetic test uses. And if we take, for example, chronic lymphocytic leukemia, one of the main markers is deletion of 17p, and patients with this deletion have a low overall survival in 10 years, so it is prognostic. And it is also predictive, as it predicts that the response will not be good to the standard treatment. Then this is an important marker for this disease. And if I take this marker, then what it really tells me is that deletion of 17 will delete this TP53, and not only deletion of TP53 is important, but also mutations of these genes are important. So if I want to find a marker for this disease, then I need to use different methods. To find deletion 17, I can use karyotype, I can use FISH, I can use CGH. And if I want to find mutations of TP53, I would do some kind of PCR of Sanger sequencing or even NGS sequencing. So it all depends on my constraints. If I have a constraint of cost and I need to an upfront cost is low, a low upfront cost, so karyotype would seem reasonable. If I need the fastest test I could get, maybe FISH would be the one to go. So there are always constraints in your specific setting that makes you opt for one or other test. So in this situation, NGS could be, for example, and this is purely hypothetical, a test that could substitute all the others. And this is specific. If I can design a test specific for my disease I'm working, then I could design a test that will search for deletion of 17p, and I could design a test that will look for TP53 mutations, and also that would look for other genetic markers of this disease. So if I use a proper designed test, proper designed NGS test, I could search for all of the things. But I would still need to consider the cost, the time, and the sensitivity, specificity of my question. So the first point I would like to address here is that there is no best test. So the solution for one disease may not be the solution for other disease. And it all depends on your local resources and what you're looking for to choose one test or the other. So what is, in fact, next generation sequencing? Next generation sequencing, it uses a little bit of previous technologies, and it depends a lot of advancements in computer sciences. We have the advances in artificial intelligence that allowed it to be developed. We had advances in cloud computing that allowed it to be developed. And especially the informatics revolutions that we all have seen and the drop in cost of computing, these all together allowed the drop in costs of sequencing. And in fact, the human genome that took around 13 years to be developed, to be concluded, and cost about $2.7 billion. Nowadays, we can sequence a genome, a whole genome for one person in a few days for a fraction of that cost, around $1,000. And also the BRCA testing. For example, one test that was one of the main tests in genetics for cancer. So it used to cost around $2,000 to $3,000, and now it's $200, $300. And we moved from a situation where we had to fish, to catch a single fish each time, and now we are in a situation that we can get a a whole of bunch of people together to catch a lot of fishes from different sizes, some very, very big ones. So uh, when we do next generation sequencing, we have some steps, and I will resume them. So if we have this information, we consider the DNA as an information, we have to capture this information, then we have to organize this information, and then we will interpret this information. Each of these steps is done by a different professional. So you have the molecular biologists working at the bench to capture this information. You have the bioinformatician that will organize the information, and we have the medical doctor that will analyze this information. This is the setting we have at Mendelics. The molecular biologists will begin with DNA extraction, then there will be a fragmentation of the genomic DNA. You have a big molecule, so you have to fragment this molecule in many pieces. These pieces will be captured by using baits specific for our targets, and then there will be amplification and fragment sequencing. So in the end, when we do the sequencing itself, we have a lot of different fragments of different sizes, and we will then read all of them, but we need to align them to the reference, the genome. At the bottom here, we have the reference genome. This is previous known, so this is the human genome, and then the fragments will be read, aligning this, solving a puzzle, putting piece by piece, side by side, so that we can know the sequence of that patient. And then we will do the annotation. What is annotation? Any difference from the reference, as is in this case here, we have 3-letter A instead of a C. So this is a change in relation to the reference. So this will be annotated. This is what we call a variant, and this variant then will be interpreted by the medical doctor. An interpretation, sometimes people ask if it is what database do you use? It's not a matter of database, it's a matter of what information can we find for this specific variants. So we need to check allele frequency, we need to check the pattern of inheritance for the disease, the mechanism of the disease, the prior information on genes, as it depends if the genes is related to the particular disease you're studying. It depends on information on previously identified variants, in silico predictions, epidemiological studies. So all of this information will be used to classify these variants. And just to give you an idea, when we do a exome sequencing, and let's say we do 1,000 exomes in the population, if each of these variants, I aggregate them, what we will find? We will find that most of the variants we discover will be singletons. What does that mean? That means that most of these variants will appear in this population just once. They are very rare. And these are the most important variants for us in terms of medical genetics. If I study the exome of just one person, to give you an idea, we'll find about 22,000 variants. And most of these variants for this individual will be common. We will have a frequency more than 5%. So probably these are not the ones that cause the disease, but the rare ones, and we'll find about 160 rare variants. These are the most important that we have to spend most of our time studying these variants. And what NGS does, it shows us when there is a difference to the reference, that is in this case of BRCA1, we have a mutation here, we have this pathogenic variant. This variant is found in about 50% of the reads. We have about 200 reads in this region. So this variant is in heterozygous state. In this other case, we have a deletion of TP53, and this is also pathogenic. And in this case, we have a large CNV, what we call a copy number variation. We have a deletion of 4 exons, and we can see this through next generation sequencing. So next generation sequencing can be used to sequence only one single gene, sequence 2 or more genes, and that is what constitutes a panel, sequencing the whole exome, all exons of all genes, and sequencing the whole genome, introns and exons of all genes. And the difference between exome and whole genome is that exome doesn't sequence non-coding DNA, and whole genome sequences the coding and non-coding DNA. But it all depends on the way the sample is prepared. We have about 20,000 genes in the human genome, and of these 20,000, about half of these genes are already described in hereditary diseases. But we are looking for cancer, for hereditary cancer. It's just a small subgroup of these genes that we're looking for. So when we would do sequencing for cancer, for example, we don't need to sequence 20,000 genes. We just need to sequence about 200 genes, or depending on the situation, different number of genes. And when you do this, for example, breast cancer, then you have the main genes that are already described for predisposition, and also for colorectal cancer. And the same for child cancer. And when we take, for example, in infancy, the proportion of cases we can find mutations, hereditary mutations, depends on the tumor. Some tumors have a higher degree of correlation to hereditary cancer, as adrenocortical carcinoma, and others have a lower proportion, as neuroblastoma. But when you take all diseases, all types of cancer, about 5 to 10% will be hereditary, and about 15 to 20% we have cases with aggregation of cancer, but we can't find the cause yet, and 80% will be sporadic. And so when we had to choose between doing a panel to doing an exome or genome, what we consider is what are we looking for. If we're looking for cancer, then the choice is to go to a panel, be it a small panel or expanded panel. Panels will give me a lower cost and higher coverage. And when can I use exome? Or if I can use exome in cancer, when I would use it? Well, I would use it for cancer research in specific settings. If I want to find new genes, if I want to study a particular family that calls my attention, then I might use a exome sequencing of this family to try to find a cause for this disease. But this is research. This is not the standard of care. The same for case control studies. I could set up a case control study to try to find new genes associated with cancer. So when I would recommend exome sequencing, when I have the possibility of a rare disease, a rare syndrome, and it's true, sometimes rare phenotypes are associated with cancer. So in this setting, I would recommend exome sequencing. But aside from that, only in a research setting. So this choice of the technique depends on my question. And as we will see in just a few moments, it depends also on my sample. Because if now I want to study tumor, I need to think what I want to study in a tumor. If I want to study a tumor, I start with tissue collection. I follow through DNA extraction, sequencing, and interpretation, the same thing. But my goal is different. When we want to sequence a tumor, my goal is to make difference in my choice of the treatment plan. That's the main goal when I want to test a tumor. And when we test tumor, we must consider heterogeneity and clonal nature of tumor. So when I show you the BRCA1 in a germline test, you have just two alleles, 50-50. It's simple. It's straightforward. But when you test a tumor, it's different. Something different appears here. We have three alleles. We have the C, it's a reference. Those are not marked. Then you have an A, and then you have an AT. So we have three alleles here. And why? Because of the nature, clonal nature of this tumor. As we all know, there is a clonal expansion in tumor and sometimes differentiation. And so in one tumor, you can have two subclones or more. And the same for treatment. When in this study here, they took 20 triple negative breast cancer patients, and they and they tested them before neoadjuvant therapy and after the therapy. And when they tested, they saw that half of the patients had total clonal extinction after the neoadjuvant therapy. And half of the patients, they found that one small subclone just grew after the treatment. And what happened? This small subclone was a minor one. It was a sensitive one, but during treatment, it acquired resistance, and then it expanded. So there was an adaptive response of this subclone. And those who work with hematology are already used to this notion. And so when we think about heterogeneity, we can think of two types. One of them is spatial. We have, for example, the primary tumor, and we have some subclones in this primary tumor, but maybe some of the metastases might be a different subclone or might have gained differences in relation to the primary one. And the second heterogeneity that we must think is about temporal. We treat today, we make the diagnosis, we begin our treatment. This tumor have some mutations, we can find them, but as we treat, as I just showed in the case of breast cancer, this tumor may acquire new mutations. There can be new subclones appearing, and for every line of treatment I can introduce, this can happen. So I must think in terms of time to repeat many times after my endpoints of treatment. So if I want to make a tumor sequencing, now I need to think about all of that. We need to be able to detect low allele fractions. This is a must when we do tumor sequencing. And we must keep in mind that we should at least find a allele fraction of 5%, ideally much less than that, and even less than 1%, because that may make a difference for treatment. And to achieve that, I need a high number of reads. I need a high coverage. And that makes tumor sequencing very different from germline sequencing. When I do germline sequencing, I don't need 200 reads. Oh, I would like more than that, but I don't need it. I can make a diagnosis much less than that. But if I'm doing tumor sequencing, now I need a lot of reads. I need a lot of coverage, so I can find small fractions. And we can divide tests in two types. So when I say about NGS testing, I can have methods that we use hybridization, and I have methods that we use amplicon. Amplicon is PCR. So essentially you are doing a PCR and after this PCR, you do the subsequent amplification and sequencing. But this is used generally for small mutations, specific hotspots in some genes. So when you have fewer targets, you can use amplicon based. When you're maybe testing for a single disease, you can make a amplicon assay to find that. But if you're testing for multiple diseases in a single assay, maybe you should go with hybridization technique, because you will be able to cover large regions of interest. So the constraints here are the sample type, what sample I'm using, what amount of DNA I have to test, because amplicon-based assay needs less DNA than hybridization. What coverage I need, because amplicon will give me much more coverage. What are the number of targets, the cost and the time? Amplicon will take less time to give me a result than hybridization. So there are many questions to be answered to define what test will you do. And which sample will I use? I can use different samples. And even if I'm talking about making a biopsy of the primary tumor, it is important to think about this heterogeneity, because I can make a biopsy of just one point and just get one clone and not the other clones of this tumor. So in order to have a representative sampling, I should do multiple points and homogenize these samples to find all the mutations of this tumor. And the other question that comes along a lot of times is about liquid biopsy. So what is liquid biopsy? It is a possibility to use circulating cell-free DNA or circulating tumor cells or exosomes. So these are three different sources of DNA that I can use to make a liquid biopsy test. And these three different sources come from different ways. So exosomal is a vesicle that is actively secreted by the cell. The nucleosomal DNA gives rise to the cell-free DNA that is circulating in the plasma. And we have the CTCs, the circulating tumor cells. These cells maybe are the ones that will give rise to metastatic disease. And so some people argue that these should be our main targets, but they're very rare and they are very difficult to select. And so it's very costly to do a test of a single cell. And most of the tests today that do liquid biopsy, we simply use cell-free DNA. It's the common ground today. So for each of these sources, we have different methods. We have different methods of acquiring this material and processing this material. And what is important is that although there is a correlation, a high correlation between the tissue biopsy and the plasma biopsy, we will find some cases, and the arrows here show that we have for example, lung cancer. And this comparison here, we have the plasma NGS did not detect two resistant mutations that appeared in the primary tissue biopsy. So sometimes you have difference between the tissue itself and the plasma sample you can obtain. And maybe this is because during the disease, you have a low burden disease, you have a lower probability of finding this DNA circulating, and you have a high burden disease, you'd have a higher probability of finding this DNA circulating. So this can be a associated with the disease itself. But one way or the other, if you do a new biopsy or you do a liquid biopsy, you probably will need, as we saw, to make multiple tests along your treatment, in order that as you make the first line treatment and you start to get resistance, you test again, you modify your treatment, and you test again, you start to see resistance, and then you change your treatment again. So this will be a cycle. And there are a lot of questions. And most of what I just showed you here is predictive or prognostic. So when we talk about tumor testing, we're essentially talking about predictive or prognostic testing. And when we talk about germline testing, we're talking about diagnostic testing. But we have also some different examples here, and this is something for the future. And one example is the question, can I use liquid biopsy to make diagnosis? So can I use NGS testing to try to make a difficult diagnosis, just as ovarian cancer is a difficult cancer to diagnose? And the answer is yes. Is it ready for the clinic? I wouldn't say so. We still cannot find most of the early cases, so it's not ready, and maybe it will never be ready, who knows. But there are a lot of people studying the use of what would be a liquid biopsy to try to diagnose disease, not only to predict response or to do prognosis for the patients. And the same goes for some situations in the clinic where we have advancements, just like carcinoma of a non-primary. We can now do something like a liquid biopsy to try to diagnose what disease is, what is primary. And we have seen some advances here. And now if we can test a liquid biopsy for diagnostic purpose for a single disease or for CUP, then there came the idea we can also try to use it to universal screening. And NGS is a technology that is being tried to make screening tests for the population, for diagnosis of cancer. If we can test for universal, any cancer, universal screening, then you'd need to screen just a few people to find one positive, and that would be good. But that depends on the specificity, and today this is still the problem. So we don't know if it will finally work, but there are a lot of people studying that and trying to use NGS testing along with traditional plasma markers to try to screen and diagnose cancer. They can find sometimes high accuracy for some cancers like colorectal or ovarian cancer. But as I said before, the problem is still that stage 1 disease has a low proportion detected. So that's not the ideal yet. And we all would like to see that get better. So there are a lot of questions with no answer. When we talk about NGS testing, we can find rare undescribed mutations in known pathways. So what will we do with that? We don't know. There's no clinical information on that. So can we use the same pharmaceutical designs to treat these patients? We don't know. There's no clinical test. If you have on-label mutations for other types of tumor, can you use the same treatment? That's not clear. We don't have information on that. What's the best testing interval if we're going to test our patients multiple times? We don't have this information. And what is the clinical utility? And that's the bottom line. And some people are trying to look at that, and the results are not yet so good. So this is just one paper And they looked for unlabeled target, 40% of the patients, that they looked for 300 consecutive patients, 40% had unlabeled targets, so approved for the tumor. 66% have off-label targets. 89% had clinical trial targets. But the bottom line, what really is important, uh, just 11% had a modification of the treatment because of the test. So the clinical utility doesn't seem to be that good. But maybe it's just because we still don't have the clinical trials to make this tool useful for us. So the main challenge today is to move through the clinical information and association studies to see what works and what doesn't work. One example that does work today is to use NGS to substitute other tests. What I said for leukemia can be used, and it's already reality, at least here in Brazil. We had a situation where to test for ALK, ROS, and EGFR, we had to have 3 different tests. The block had to go and come back 3 times, and you would spend 3 months to get all the results you needed. So pharmaceutical companies joined themselves in a consortium, and here we can see that NGS indeed can find ALK fusions if they are properly designed. And so I was saying that pharmaceutical companies made a consortium, and now they take just the sample one time and do an NGS testing that substitutes the previous ALK, ROS, and EGFR different tests. So in about 14 days, 2 weeks, now the patients can have their results. And all these questions are being studied. A lot of people are studying this. There's a big NCI trial, and they release a previews of their results. And one thing that showed my attention is that although they have planned for a 14-day turnaround time, they were getting more than 30 days to get the results back. And that is one important issue for people who works with cancer. You need the result as fast as possible so you can change your treatment. So my point here is that NGS sequencing is a tool. It's essentially a tool. And my taking home messages are, NGS is not one method. NGS is a general name that refers to different types of massive parallel sequencing. It's not necessarily better than any other method. It all depends on your available resources and your clinical question. And to test more genes is not necessarily better. So it's not better to test a big panel. I need to test what I need for my disease, for the disease I want to test. The number of genes depends on the disease you want to study. And if you need tumor sequencing, standard NGS technique for hereditary cancer probably will not work because it will not be capable of detecting slow fractions. NGS testing for germline is our goal is to achieve 20%, between 20 and 50 or 70% of allele fraction. If it goes more than that, probably it won't be detected. You need to make sure that the NGS tests you select have the appropriate baits and coverage needed for the tumor of interest. So if you want to test hematology, you need a test that will have TP53, that will have RUNX, that will have the genes associated with hematological malignancies. If you test a tumor and a pathogenic variant is found in a gene that could also be inherited, then you should also test germline. I didn't explain this before, but in short, Every time I find a, a pathogenic variant in a tumor, and this variant is in a gene that could also be inherited, then I should consider tests. For example, if I test a child for a brain tumor and I find a mutation in TP53, I should consider this child might have Li-Fraumeni syndrome. So I should do a germline testing to find if this TP53 mutation was inherited or not. So this is one special situation. And Mendelics, well, Mendelics, just a few words. We are one of the major labs in Latin America, based in Latin America. And what we do is just we test constitutive DNA. So we don't do tumor testing. We test for rare disease, we test for hereditary cancer. I thank you for your time, and I'm sure we have plenty of time to get some questions.